All right. So my goal for you today is for you to be able to mainly differentiate between the three major types of membrane receptors. And this is going to be kind of a drawing heavy day. We've got like three different processes to draw out. So if you are a color coding person and you don't have different colored pens, pencils, highlighters, I've got my colored pencils out over there. Feel free to grab a couple, because we've got three major like protein receptors, and there's like a lot of different stuff going on. Before we get started, does anybody have any questions? No? All right. Well, yesterday we talked about like overarching, like this is what cell communication is. We had some examples, and we talked about three major types of external cell signals. We remember what those are? Yeah. Contact, local, and long distance. There are cell signals where we've got two cells actually physically touching one another. We've got a cell signal sent across an extracellular space, but not too far. And we've got signal sent from head to toe within an organism. And those are the three types of external signals. Today, we're going to talk about what happens when those signals actually reach another cell. So that is the goal for today. I forgot that my markers were charging, so I have to fix that. So starting us off, let's see if this will work. Haha, basics. So, when a signaling molecule will bind to a receptor protein, all of these receptors today are proteins, that protein will change shape. If a protein changes shape, what else does it change? Function. This change in shape causes a change in cell or a protein function. And the type of, and whether or not that signal is received by a cell wholeheartedly depends on whether or not that cell has the receiver or the receiver gets that message. So it's like nobody can call you if you don't have your phone on you, right? Nobody can contact you if you don't have a means of contact. Same thing with cells. Cells can't receive messages if they don't have the little receptors on their membranes. Drop the mic pack. All right. So, a signaling molecule will bind to the receptor protein. That receptor protein will change shape, and then that change in shape will result in what we'll learn about tomorrow with signal transduction. The reception of the signal, like I said, depends on whether or not the cell has the receptor. Cell can't receive the message if it doesn't have a way to receive the message. So everything depends on whether or not they have the receptors on the outside. Some quick terminology. Signaling molecules are also called ligands. Now, just like our proteins are very specific in shape, right? Shape determines function. The shape of the signaling molecule has a specific place on the receptor. Whoop, yes, please. Site. I always forget that it kicks me off once I hit enable. There we go. Site on the receptor and attaches at that specific site. Just like when we talked about enzyme specificity, receptors are specific as well. So we've got our specific enzyme ligands. We've got our specific receptor ligands. A ligand is just a molecule that binds to another molecule. That's why we can use ligand in both enzymes and receptors. Generally, like I said, this causes the receptor to change shape. It's on here twice because it's just that important, right? That change in shape is what triggers the next step in the pathway. So, I won't make you write it twice. if it wasn't important. So, signaling molecules have a specific shape. That shape matches a site on the receptor. That's where it attaches. Just like the active site on an enzyme, we've got a special binding site on a receptor. Signaling molecules act as ligands. Ligand is a general term. Ligand is a general term. It is just a molecule that is very specific that binds to another. So that other is typically larger because it has to, fit inside of it. Generally, the binding of this molecule causes the receptor to change shape. That change in shape kicks off what we'll talk about tomorrow. The receptors we are talking about for the first bit of this class are all membrane proteins. So they are proteins that are embedded on the outside of the cell membrane or embedded in that cell membrane that can receive signals outside of the cell. The very last slide today, we'll talk about how there are some receptors inside the cell, but mainly we're focused on the outside because we're looking at cell-to-cell communication, how cells talk to one another. So these are signaling molecules. Questions so far? All right. I'm so tempted to pay for premium just so I can access all of these other colors. I'm tired of writing in just one color. All right. So, there are, like I said, 3 major types of membrane receptors. These are the 3 that we're going to talk about today. The first one is a G protein coupled receptor or a GPCR. I usually just say G protein because GPCR sounds like alphabet soup in my brain and I get them mixed up. So that's the first one we're going to talk about. We're also going to talk about receptor tyrosine kinases, and ligand-gated ion channels. So these are our 3 big classes of membrane proteins. Most membrane receptors, almost all receptors fall into one of these categories. Almost all of these receptors fall into one of these categories. And we'll talk about it in a moment, but these G protein coupled receptors are like the largest class of membrane receptors. So I've got my drawings on my little notebook. All right. So these are our 3. G protein coupled receptors, receptor tyrosine kinases, and ligand-gated ion channels. Those are our 3. So naturally, we're going to work through them one by one. Starting with G protein coupled receptors. G protein coupled receptors. Like I mentioned, this is the largest family of receptors. We found over 800 types of G protein coupled receptors, which is kind of crazy. So we're going to draw out the general setup of a G protein. So the receptor and the G protein are separate entities. That is why it's a G protein coupled receptor. So we've got a receptor, we've got a G protein. The way this works is we've got our cell membrane. We've got our cell membrane, and we've got our G protein coupled receptor. We've got our G protein coupled receptor. We also have our actual G protein. And I'm going to abbreviate protein with that little P with the line through it just for cleanliness' sake. That G protein can move along the cellular side of this membrane. So remember we said membranes are fluid. We call it the fluid mosaic. Stuff can move around. This G protein can definitely move around. It can float this way, back, forward, this way. This G protein is mobile. It's not like attached to anything. And it helps that it's only on one side of the membrane. It's only on the cellular side. And this G protein can be active or inactive. When it is inactive, it is bound to a GDP, like ATP, but it's guanine triphosphate instead of adenine triphosphate. But when it's inactive, it is a GDP. So the less energy, the di instead of tri, is the inactive form of that G protein. And if there is a GTP, it is considered active. We've also got one more player in here, and that is this enzyme. We're going to make it like a little Pac-Man. This is an inactive enzyme. We're just going to finish off this membrane too. This is our general setup. This is kind of the default state when nothing's going on. Our G protein is inactive, our G protein coupled receptor is inactive, and our enzyme is inactive. We are going to work through the steps of what happens as the system becomes activated. So, questions here. All right. So, first things first, a signaling molecule is going to bind outside of the cell to that G protein coupled receptor. So, we've got our ligand coming in to that G protein coupled receptor. So, we're going to bring that receptor back. And we are going to have a ligand bind to it. G protein coupled receptor. When this happens, when we've got a ligand, it allows that G protein to bind. We're still not messing with this enzyme yet. When this G protein binds, it is going to displace the GDP that was on here. So we're going to lose the GDP. And we are going to now bind a GTP. This G protein now has a GTP. That means it's active, it's excited, it has energy. GTP has more energy than GDP. Our enzyme over here, still inactive. So, we had our inactive states, and then we had our signal, our external signal from some other cell that comes in and activates our G protein coupled receptor, which then allows the G protein to bind. So when this ligand binded, it changed the shape of the G protein coupled receptor, which allowed the G protein in, kind of opened the door for that G protein. And when that G protein binds with the receptor, it actually dislodges the GDP and allows a GTP to take its place. Doing okay so far? All right. So we now have an active G protein. All right. I've got those little lines around the G protein to show that it's active. All right. So now that that G protein is active, it will move out of the coupled receptor and over to the enzyme. So our G protein coupled receptor, that ligand is actually gonna leave. That binding is reversible, so it can just kind of pop off whenever it's done. And now the activated G protein can slide along that membrane over to the enzyme. This activates the enzyme. That active enzyme can then trigger a cellular response. This bit is what we're going to talk about, tomorrow. We're going to talk about tomorrow. Messed up my tenses there for a second. So, that G protein, the activated G protein, remember it's got a GTP on it because it's active. We'll then slide over, and now that it's active, it can activate our little Pac-Man enzyme and trigger our response. All right. You doing okay? We got one more step in G proteins. All right. So our ligand's going to leave. It's going to send our GTP activated G protein over to our enzyme. Enzyme's now activated. It's going to result in some sort of cellular response. But now that we have our response, that activity from the activated enzyme takes energy, right? So it's going to take that GTP and hydrolyze it to GDP to use that energy that's stored there, just like we do with ATP. So our G protein coupled receptor still just chilling here. It's not doing anything right now. What's going to happen is that G protein is actually going to have a GDP again, and we're going to have another random inorganic phosphate chilling over here. And our little Pac-Man enzyme is now going to be back to being inactive. after it sends this away and releases this. This very much looks like the first picture we drew together. Because it is. We're back to ground state. Now another ligand can come in, bind to this, and set the whole process over again. My membranes got a little more tilted as we went through, but it'd be like that. How we doing? All right. Just kind of a random thing that I haven't really mentioned so far. I've just been using the word enzyme over here, right? Activated and inactivated enzyme. That's because, like I said, there are like over 800 of these G protein coupled receptor like types. So there's not like a specific enzyme that is activated by this G protein. There's a bunch of them. So this is just like a general like skeleton idea of what a G protein coupled receptor does in a cell. That's it. Just a lot of kind of like intermediate moving back and forth, send a message, activate, so on and so forth. But this is like one of the most common ways that our cells know how to receive signals. I find that pictures tend to help. All right. Questions about G protein coupled receptors? No, maybe so. All right. We got one of the three down. That was the most complicated of them all. We kind of started out with the big one. We're going to work our way back down. So after G protein coupled receptors, we are going to talk about receptor tyrosine kinases. A receptor tyrosine kinase is a type of kinase. A protein kinase is just a general term for an enzyme that catalyzes. Remember, enzymes are biological catalysts, so catalyzes the transfer of phosphate groups from ATP to a protein. So a kinase, just a general kinase. What it does, this is a protein kinase. It will take an ATP and then transfer. Actually, I'm going to draw out ATP. We've got our adenosine, phosphate. And we've got another random protein. What that kinase will do is it will take one of the phosphates off of ATP and move them to a protein. So we now have a DP and our protein that has been phosphorylated. You don't need to have that picture down if you don't want to, but that is the general activity of a protein kinase. A receptor tyrosine kinase is a receptor protein, so it is in the membrane, it responds to a signal, and it transfers phosphates from ATP to kinase, or tyrosine, sorry, it's tyrosine. Tyrosine, protein, yeah. So this is our general kinase system. We're going to take a phosphate off of ATP, chuck it on a protein. Receptor tyrosine kinases is a specific type of protein kinase. So protein kinases are general umbrella. Receptor tyrosine kinases are specific. So like if kinases are like cookies in general, receptor tyrosine kinase is a type of cookie. So like chocolate chip. Yes. Nope, that's just helping me explain to y'all what this sentence means, because catalyzing the transfer of phosphate groups from ATP to proteins can get kind of technical. All right. So, here is the process of activating a receptor tyrosine kinase. So, receptor tyrosine kinases, in its inactive form, is a monomer. It's just one. So, it kind of looks like this. We've got our little binding site, and we've got 3 tyrosines. This is a receptor tyrosine kinase. It's going to resemble receptor tyrosine kinases for us today. This is our inactive monomer. This is going to be embedded in a membrane also, like up here. Like that. So the receptor bit is outside of the cell, and the tyrosines are inside of the cell. If that helps orient you in where we are in our cells right now. So these are our wonky little receptor tyrosine kinases. All right. Cool. So, when a signaling molecule hits that little binding site, 2 of those receptor tyrosine kinases will actually bond to one another to form what we call a dimer, which is just 2, it's like polymer but 2. So monomer, polymer, dimer is like 2 of them. So what happens is if we have our 2 receptor tyrosine kinases, we're gonna draw our membrane in here. If our ligands bind, then what happens is we get our dimer, which I have a hard time drawing sometimes. There we go. We've still got those ligands in there. They got a little bigger, but they don't really get bigger in real life. That's just the way my picture happened. This is our dimer. These are still tyrosines. Okay. This dimer has activated tyrosine regions but is not fully activated. So the change in shape has kind of activated these little regions here. But the protein itself is not fully active. So what happens next? The receptor tyrosine kinase does what a kinase does. It hydrolyzes ATP and transfers those phosphate groups to those little tyrosine regions. So, our dimer is the activated form of. Yeah, okay. This is the not fully activated. There we go. And then after we've phosphorylated our ATP, then we have fully activated the molecule. So while y'all write that down, I will get my little drawing set up. All right. There we go. So, the way this works is our receptor tyrosine kinase, once it's not fully activated but kind of activated, will take 6 ATP and they will hydrolyze it or take those phosphate groups off and transfer them to our tyrosines inside the receptor tyrosine kinases. The reason we need 6 ATP is because each monomer has 3 tyrosines, so in total we have 6 tyrosines. This is our. Don't know what's going on there. This is our fully activated phosphorylated dimer. I always make my phosphate groups different colors so I know what they are, but that is just me. All right. So now we have a fully activated receptor protein. What happens next? Button on the side. So we've gone from a inactive monomer to a dimer to a fully activated phosphorylated dimer. It's a lot of weird drawings today. So now that our molecule is fully activated, it can lead to our cellular response. So we can take those activated tyrosine regions and we can have them act in different transduction pathways. So we've got our, one more time, fully activated these ligands are still in here because it's activated. What happens is that relay proteins, I'm just going to make those kind of wonky shapes. These are relay proteins. They are going to bind to our activated tyrosine regions, and then they are going to give off our cellular responses. What's really cool about this is that we can have multiple cellular responses from one receptor, because we can have multiple relay proteins bind to those activated tyrosine regions. Questions about receptor tyrosine kinases. All right. That is our second type of membrane receptor, our second class of membrane receptor. I got one more for you, and it is the simplest of them all. I'll give you a second to get this picture real quick. Okay, so we've talked about G protein coupled receptors. We've talked about receptor tyrosine kinases. Now we just have ligand-gated ion receptors or ion channel receptors. What these do is they act as a gate that will open and close as shape changes. So, default state. We've got our receptor. This is the outside of the cell. This is the inside of the cell. And we've got ions. Those ions cannot pass through the cell membrane, can't diffuse through the cell membrane. So we have to control whether or not they are let in or not through this gate. So what happens is that when our ligand binds to our receptor, so we've got our ligand coming in, it will actually open up this protein. It will cause a change in this protein that lets those ions move across the membrane. Quite literally, ligand gate for an ion channel. That ligand binds, changes the shape of the protein so that it is open, allows ions through. But it just gets bigger every time I draw it. Oops. The second that ligand leaves, our gate is closed again. Still got ions inside, but they can no longer pass through that gate. I said that was the simplest one. So that's all we've got for ligand-gated ion channels. It's a little gate. You got the little key. I still have one more slice for you. Sorry. We're almost done. I promise. So all of the membrane, or all of these receptors that we talked about today, are membrane proteins. They are embedded in the membrane and receive external signals. But cells can also receive signals inside if those signaling molecules can pass through membranes. Hormones are able to freely diffuse through cell membranes. Hormones are able to just kind of diffuse through lipid bilayers. So, if they can just diffuse through our cell membranes, we need a way to receive those signals that isn't embedded in the membrane because it could just go around it. So, what happens is we have some receptors that are either found in the nucleus or just kind of free floating in the cytoplasm. We're not going to talk about those specifically. Like, we're not going to go through like a bunch of drawings with different internal receptors, but I wanted to, tell you they exist because they do, and they're really cool. So this often leads to turning on or off genes. This is usually what these signals are for, turning on and off particular genes. Of course, I was still on highlighter. So these hormones, the intracellular signals flow through the membrane. Once they're through the membrane, they can reach their receptors inside the cell. And once they reach those receptors inside of the cell, it usually leads to like directly controlling which genes are transcribed and translated. But some of them have a few other random effects. But questions about cell receptors? All right. That is what I have for you today. There is no exit ticket because we drew a lot today. And we only have like 5 minutes left. So, tomorrow we are going to talk about signal transduction pathways, what happens after reception that leads to our cellular response. No, you got a couple minutes to kind of chill out. If you didn't finish that exit ticket from yesterday, I highly recommend you use this time to do that.